So I will spend like maybe 10-15 minutes of the lecture talking about very basic concepts, and then we will go to the part that regards more specifically the methods that we use to analyze this kind of sequence, to align this kind of sequences. So the building block for all living organisms are cells. So cells is the main component of each living organisms. And somehow there are two main kind of cells, and the simplest one are prokaryotic cells that are the very simple one, and then we have the eukaryotic cells. So we are done of eukaryotic cells. They are. Do we have a pointer? Those cells have a core, and this core is called the nucleus. Inside the nucleus, there is the genetic code for the cells. Somehow there are some other compartments that are also useful for the living of this cell, for the evolution of these cells. So inside the cell we have many small particles that are bounded by some membranes. The most important ones, and the one that we are focusing on, is the nucleus. The nucleus contains the genetic material, and inside the nucleus. The red one, thank you. And inside the nucleus, there is the transcription and RNA maturation, and we will see a little bit about this. Another part that is also important is mitochondrials, because they are devoted to the production of energy inside the cells. And inside mitochondria, there is some other DNA. Somehow these mitochondria were, at the very beginning, they were bacteria, and they came inside the cells, and they found a good living habitat. So they started living inside the cell, and nowadays all the cells have those mitochondria. So these small organelles, many of them, with their own genetic material that produce energy. So the first definition that we will give are regard the genome. So the genome is the complete set of sequences that make up the genetic material of an organism. So inside the genome there is everything, is written, everything that the cell needs to know. So it's the program of the cells is written in his genome. And the parts of this genome, so this genome, you can think at it as a very long strand, as a very long string. In case of humans, this string is composed of 3.something, 3.2, 3.6 billions of letters, a long string made up with many letters. And parts of these genomes are called genes. Genes are functional pieces of this genome, and they express themselves. So their activity is to produce a molecule that is called RNA, and we will see this in the next slide. So gene is the part that is devoted to the production of proteins, and proteins in our body does almost everything. So this is the most important part, and the part of the genome that the part of the genome that expresses those proteins is very small. For example, for humans, this sums up to a very small fraction of the whole things. So, and this is the one in which we are usually interested in. The genome in itself is divided and is composed in chromosomes. So those chromosomes are the units in which the genome is organized. And different organisms have a different number of chromosomes, and the chromosomes are duplicated in every organism in such a way that there is a fault tolerance. And the chromosomes are included in the nucleus, and they are present, as I said, in a different number in different organisms. As I said, there are two main types of genomes. One is in the nucleus, and it is composed of, as I said, of 3 billion nucleotides. So we can think that every single nucleotide as a single letter, and the alphabet on which it is based is composed of only four letters. And then we have the mitochondrial genome that is much, much smaller. As I said, it was initially a bacterial genome, so it's completely different in shape and structure from the nuclear genome. It's a round, it's a circular DNA, it's very small, it's only composed of 16,000 nucleotides. And the main purpose of this mitochondria is to produce energy. So, as the genes is what you inherited during evolution and during life. So this is the functional units, and the genes are localized in specific positions within the sequence, and inside the genes there are all the information that are needed to the program that is needed to code for proteins. There are also some genes that do not code for proteins, but somehow they do the same process, so they do transcribe, they do produce RNA, but it is a different molecule. It's called non-coding RNA, and somehow this non-coding part is a very large part, and it's also very important because this non-coding RNA has effect on the genes. So the action of transcribing, the fact that the gene produces RNA, and this RNA is then transformed in a protein, is somehow guided by non-coding RNA. So the activation and deactivation of genes can also be done, and modulation of the quantity of RNA that is produced by a gene is somehow guided by non-coding RNAs. And this process, this fact that the gene activates and produces RNA is called gene expression. So the main gene expression. As we said, all the genes are on chromosomes, and the chromosomes are inside the nucleus. So the DNA, that is the main, as we said, the main program that we have within the cell, is organized on chromosomes, and those chromosomes are inside the nucleus of the cell. In a eukaryotic cell, they are located, and everything that is written there is what we bring with us. The very interesting thing is that a very little variation in DNA. So let's say we said that this DNA, we can consider it as a string with 3 billion and more nucleotides on that, and even a single mutation in one of those letters can produce very bad effects. So there are many genetic diseases that are due to the mutation, so to the variation of a single letter within this long string. So what we are interested in many times is actually to look at those variations, to find where those letters are different from a reference genome, from the standard, let's say from the normal situation. So the genome, all the genetic material, as I said, is composed of nucleic acids that consists of nucleotides. So the DNA is a sequence formed of those 4 nucleotides, A, G, C, T. And we will not care too much about the meaning of this. What is important is the fact that somehow these are the building blocks. And the first attempt to sequence the human genome was tried out at the end of the 90s. There was a huge project that was founded by NIH, that is the National Institute of Health from US, and they wanted to sequence the entire genome of a person, of a human being. Before that, the existing solution that we had were somehow due to Sanger. Sanger is the only person that has 2 Nobel Prizes, and he got these Nobel Prizes for finding a technique that was capable to sequence thousands of nucleotides. So his technique was able, given a small piece of DNA, long 1,000 bases, to find out the letters that were inside. Of course, if you want to sequence a genome that is 3 billion letters, you cannot use a technique like this, because if you try 1,000 at a time, it will take too long to get the entire sequence. So NIH was pushing forward research, trying to get new techniques, and somehow the technique that was developed was based on parallelization. So the easiest thing to do was to take this big molecule, DNA molecule, and to cut it in small pieces. So every single piece was then sequenced in parallel on a machine, and actually the machine was sequenced in parallel on those machines. And actually, to build up again the genome together, they used a very big machine. They used a cluster of computers. At the time, they were alpha processors. They were digital processors. And actually, there was a competition between a public consortium and a private project that was done at Celera Genomics. So this private company that was funded by Craig Venter was actually competing with the public institution, the public national labs. And somehow they ended up first. So they were the first to produce an almost complete copy of the human genome. Actually, it was the genome of Craig Venter. And so this was published in Nature and the next day on Science. And this was the first draft reference genome of the human beings. Of course, talking about the genome of a human being is somehow ill-posed, in the sense that we are all the same, but we are not completely all the same. The difference between my genome and the genome of every person in this room is about 5%. So this is the natural variability that there is among us, and this small variability, of course, makes us very different, in the way in which we appear, in our phenotype, and also in the diseases that we carry with us, the genetic diseases that we might carry with us, and the diseases that we will develop during our lives. So, so the interest was actually to get a reference. So the interest was to get a genome on which we could make a comparison. And this is still the case. After this, at the time, the sequencing of this genome cost something like $100 million. But nowadays, this pushed the technology to get new machines faster and cheaper. And nowadays, we can say that we can get a complete genome of a single person in, let's say, 11 days, less than 2 weeks, and for a cost that is approximately in the order of €1,000. So the prices have slowed down, have gone down very, very much. So the prices, now it's very cheap to do the complete sequencing of the genome of a single person, and it's also somehow fast. Not very fast, but it's somehow fast. So this poses new questions, because now that we can have all this data, we can have the genome of every single person in a very short time, then we need to analyze. So we will need algorithms that are very fast and can determine the differences between the genome of this single person with respect to the normal genome. As I said, we are not exactly all the same. There is this slightly difference that is called natural variability. Of course, natural variability makes the color of my eyes different from the color of the other people. The hair, for those that have, different in colors and shape and whatever. And so this is natural, and there is nothing wrong with this. But as I said, there are some small variations. There are very small mutations that can make things go very weird, very bad. So, as I said, in the case of genetic diseases. And so it's very interesting, as I said, to find novel techniques to analyze this data. The data that are now available is incredibly huge. So they are all publicly available. They are stored in databases, and the access is public, and you can download this data and try to do your analysis. And of course, a slightly advancement in the computational complexity of algorithms can produce very big improvements in the computing time that we need to analyze the genomes. The first step that we get is always the same. So we take those small pieces, as I said, when you want to sequence the genome, you have to cut it in very short pieces, pieces, and many short pieces, and you have to sequence. And then you have to match with the reference genome. You have to build it up back again. And this is the first step. So the sequence alignment is the basic for everything that you want to do after this. So that's why people is so much interested. And the research is still active after so many years. Also because this is a period in which technology is somehow pushing science. No? There are some periods in which science pushes technology. For example, if we think about the mobile phones that we have in our pockets, science gave the results to produce those mobile phones maybe 100 years ago. So science was somehow pushing technology. So the theoretical results were already available to build very small devices, very small components. Nowadays, we are in the opposite situation. Technology is pushing very much the science. So we have those machines that produce this data, but somehow we are not really able to understand what is the meaning that is inside this data. And so we need to develop novel techniques for the analysis. So somehow there is also DNA is composed of a double helix. So there are two strands. So each of these is called two strands that are anti-parallel and go in the opposite direction. Since the nucleotides are somehow pairing with a specific pattern, we find that every time that there is, let's say, a C on one strand, we find a G on the other strand. And every time that we find an A on one strand, we have a T on the other strand. So the way in which they match from the two strands, they match from the two strands is always the same. So the two strands are somehow complementary. And you can read one and you can get the other one back again. So every time we will only look at a single strand, so we will only look at a single sequence, because the other one we can obtain very easily. So every time we will talk about sequences, we will talk about strings, DNA strings, we will talk about a single DNA string, and we will look at it. As I said, the complete genome is for humans is 3 billion base pairs. But and as I said, they are organized in chromosomes. Chromosomes are somehow detached each other. They live all together inside the nucleus, but they are single pieces. When we look at the genome, somehow we look at it as it is a single long string. So what we get is a long string, and somehow if we were able to write this entire sequence of the human genome in a font like Times New Roman size 12, we would end up with something that is 5,000 kilometers long. And every human being, as I said, has its own individual genome sequence that is slightly different from the other, with the exception of homozygotic twins. And somehow there are many differences between the genomes of two different individuals. The one that I was pointing out are polymorphisms, and so there are positions, so single nucleotide in the sequence that contain different nucleotides. And this can be either natural variability, so something that only makes some slightly difference in two organisms, or it can be deleterious. And there are some other forms of differences that can be, for example, deletion. So one of the bases, one of those letters is deleted inside the DNA, or it can be added. So there is an insertion. One letter is inserted inside the long string. Or there can be permutations, so some pieces are permutated along the string. Or there can be inversions, some pieces are inverted inside the strings. And all of these, some of these don't do anything bad, some others are of interest because they produce some very deleterious effects. And then there are microsatellites that are those repeated nucleotide sequences in which the number of repetition is different between one individual and and in another. For example, when they do forensic analysis, when they want to match the DNA of two people, of one people from a database to see if he's guilty of some crime, they actually, what they do, they analyze this STR, that is this short tandem repeats. So they count the numbers, and then those are unique for every single individual. And a very single number of those, like a few tenths of those, can actually characterize a single individual. So the probability that two different persons have the same numbers inside the STRs is very, very low. And so we can say that is unique for every individual that is on the Earth. So the genome is very, very different inside, and of course it has evolved during the years, during the evolution. So this is a trial and error procedure. So in evolving, and so each cell produces a new cells, and in this process, somehow there are some small errors. These small errors, some of those small errors do not give rise to anything bad, so they are kept, or they may introduce a characteristic in the cells that makes it more suitable for living in a certain environment, and so they are kept. Whereas some others inhibit the cell from living, and so they are dead. So in this evolution, the genome little by little is evolving, and even in a single person during his life, in the process of when new cells are produced, there are errors in the DNA, and those small errors accumulate during the life. And this is somehow the reason why we think that some diseases arrive only at late ages. No, they do not are there. Even if they are genetically based, they only occur at a very late stage of the life. And so the genome size is very, very different, and somehow is not telling anything about the complexity of the organisms. As we can see, there are some flowering plants that have genomes that are much bigger than human genome. But this does not mean that the organism is more complex than ours. So we can say that our genome, if we want, our genome is comparable with the one of tomatoes, but we are very different from tomatoes. And this is probably due to the fact that our genome is capable to produce molecules that can interact in very complex ways. So the way in which the genome is producing proteins and the way in which those proteins are interacting with each other will make the difference between the different organisms. And the more complex is the organism, more complex will be the interaction among those molecules. So what is written in the genome? The genome contains the information necessary to necessary for the operation of the entire organism. And of course, the DNA in every single cell, in every different tissue, is always the same. But the way in which it behaves is completely different. So if we think about a neuron, and if we think about a cell in our skin, they have exactly the same code. So they have exactly the same information. But the way in which they use this is completely different. So during the life of the cell specializes for some specific purposes. So this process, as we said, there is this process that is called the gene expression, in which the gene transcribes and translates, and this gives rise to a new molecule that is the protein. And this is the central dogma for all the biologists. So we have those coding genes inside the genome that represent a very small fraction of the entire genome, and they transcribe and they code for proteins. The protein coding genes transcribe and they produce this new molecule that is called mature RNA. And after another process that is translation, this is translated in amino acids, so in a different molecule that is protein. This string of amino acids then folds up because it's subject to forces, and it produces something that is called, it produces a molecule that is the protein, the folded protein. If something goes wrong in this protein, in this process, then then, for example, there is a single mutation somewhere in the gene, what can happen is the protein does not fold. If it does not fold, it will not behave in any way, so it will not do its work. And somehow the protein structure is related to the protein function. So this is a central dogma. So proteins that have a similar structure, then they will have a similar function, and they are at the basis of every single every single action that happens in our body. So as we can see, the non-coding part is very large, is very different in different individuals. And as we see in humans, it represents nearly 100%. It's about 98, 97%. So the part that is not coding for proteins. Okay, so I think that we can now switch to the part that talks about the algorithms. So let me take the other presentation. So as we have seen in the first part, the alignment is a way. So aligning two sequences is a way of comparing those sequences. And comparing a sequence is very interesting because the similarity between those two strings may tell us something about the function of those sequences and of those molecules. So, and the similarity may be a consequence of a functional or structural or evolutionary relationships. So when the organism evolved and they started to differentiate each other, then they maintained some building blocks. So there were some particular parts that were maintained during the evolution. For example, all the genes that code for proteins that keep the structure of DNA, they are maintained unaltered during evolution. So you can find those genes in very old organisms. So the organism in the time they evolved and they differentiate. And if you go back again to the beginning, you find that some parts of this string, of this DNA string, is maintained among the organisms. So trying to understand which are the parts that are maintained, so finding similarities among the strings in these different organisms is very interesting. And the main thing that we do is to compare. So when we try to compare two strings, as in this example, we try to align those strings, we get something that is like this. In these examples, we have an amino acid, so it's a protein sequence in which we have those letters that are the letters that are representing the amino acids. They are 20, and when we try to align, we find that somehow, even if they do not look exactly the same, they might have something in common. And this is for example an alignment that is produced by Classe-Salvo. This is quite intriguing because what you see looking at the sequence does not seem to tell you anything about the similarity of those two strings. But we will see that there are techniques that can tell us how close are those sequences together. So even if sometimes you don't find exactly the same, amino acid, you are still finding something that is interesting, because somehow those amino acids are not completely apart. So one of them can substitute another, and this will not give rise to anything suspicious or deleterious for the organism. So there are some main objectives for the comparison. The first is the molecular phylogeny, and we will talk about this a little bit more in detail. And as I said, we are not talking about things that are created ex novo, but we are talking about things that are evolving. So those things are little by little evolving, and they are introducing modification among each other. So what we are interested in is to understand which are the things that they have in common and which are the things that are actually that are different. So from a molecular point of view, what we are interested is to build up a tree that illustrates the distances and the evolutionary relationship among the molecules that we are analyzing. We are not looking at morphological characteristics of those organisms, but we are looking at them from a molecular point of view. So we are looking at those molecules that can be DNA, RNA, proteins, and we want to try to understand what they have in common and what they have in different. Another motivation for aligning sequences and to match those sequences is to understand the evolution of individual genomes. So we want to understand what is conserved among the organisms. For example, you can have different flies that they all started from a common organism that was back in the years, and then they evolved differently. And in this evolution, the slight modification that were introduced in the genome give rise to characteristics that enable them to live in different conditions, in different ecosystems. So if we understand what is different, then we can understand, for example, why one fly is capable to live at very high temperatures, whereas another is not able to live at such temperature. And this is also used for studies in population genetics to understand how the human, the genetics of population is changing in the years. And of course, a way to study novel organisms is to compare with the existing ones. So as we said, we sequence a new genome. This happens every day. At the moment, we have something like 1,000 genomes that have been sequenced, and every day there is a new sequence, a new genome that is sequenced from a different organism, some strange bacteria or some strange fly or some strange animal that we find. And somehow, how do we try to understand the code that is inside this? We compare with an existing one. So what we try to do is to understand if there are parts that are in common with some other organism, and if they are the same or are somehow homologous, we can try to infer what is the function of this part of the DNA. So we can try to find the genes that are allocated on this genome, and then to try to understand the products, so the proteins, what they do, trying to find the similarities among the strings, among the strings, the sequences. There is a concept that is very okay. There is a concept, I'm sorry, for me it's very early in the morning, so I get a little bit confused about the time, so 10 minutes. So yeah, let's go a little bit further. And as I said, there is a concept that is very interesting. That is, there are two concepts that we need to understand. One is similarity, and one is homology. So this is slightly different. This is slightly different concept. So we can have that when we compare two sequences, then we can find that they share a certain degree of similarity. So if we look at the two sequences, we can find some places of some parts of those strings of those sequences that are quite similar. So the letters that you read are almost the same. And of course, this similarity can be measured in different way, and we will see which is the way in which we measure the distance of those strings, of those sequences. And the similarity between two sequences may constitute a structural and functional similarity, because as I said, if you have a genome of an organism that you don't know, you try to find the pieces that match with some other organism to infer the functionality of this piece. And the second concept is the homology. The homology means that two sequences have a common evolutionary origin between them. So they are not similar. They might be similar, but this is not always the case. And somehow, but they have a common ancestors. So this means that there is an organism, a very old organism, from both the two new sequences have evolved. And of course, the homology is a little bit more tricky to detect, because what we want to see is the fact that the molecules that are produced, for example, by two genes, are going to do the same function. So there is an intermediate step that is a hidden layer. So you don't see exactly a similarity between the two sequences, but you find an homology. This means that what is produced, the molecules that are produced, are going to have the same function and structure. This might be the same because we have, for example, humans have 20 amino acids, but some of those amino acids can be substituted. And when you substitute one with the other, what happens to the structure, to the final structure of the protein, might be that there is nothing really different. There is also another fact that we have to take into account, is the fact that somehow when we transcribe, when we translate from RNA to proteins, the nucleotides are taken in triplets. So every 3 letters, they are read with a window of 3 letters. And those 3 letters sum up to an amino acid. So the letters in the alphabet are 4. In the nucleotide alphabet, we have T, C, G, A. Okay? So if we take 3 of them, we end up with 4 times 4 times 4 possible combination. That adds up to 64. Now the amino acids are only 20. So this means that different triplets can give rise to the same amino acid. So different combination might give rise to the same amino acid. This is an important thing. So even if you don't see the same letter in the DNA, now, when you look at the strings of DNA, then you can end up with the same amino acid. And so this gives rise to this concept of homology. Of course, you can compare 2 sequences, but you can also try to compare more sequences together. So you can have a certain bunch. So you can have, for example, 5 short amino acid sequences, and you can try to align them all together, to understand which are the common parts and which are the different. So we will start analyzing the techniques that are used to compare 2 single sequences. And then we will go to the techniques that are used to compare multiple sequences all together. So a bunch of sequences, you take them all together and you try to find what they have in common and what is different, let's say. Do you try to align them? And then we will go to a next step. So we will try to understand which are the algorithms that are used for this massive sequencing technology. So as I said, nowadays you do not compare very small sequences, but you compare complete genomes, so, or at least complete exomes, so the coding part of the genes. So when you have those when you try to sequence these very long strings, this very long sequence, as we said, you cut it in pieces, in very small pieces, and then you try to match with the reference genome. So this is a completely different technique, and we will try to understand which are the mathematical and computer science concepts that are beyond this. You are okay? Okay. So let me make one simple example and then we stop, okay? So let's see what means aligning strings. So we have a pair of strings and we want to get an alignment of them. So let's say that those 2 strings are different. They are based on the same alphabet, of course, but they are different in length and in the composition. So the letter that compose. And we want to establish a similarity between those 2 strings. So What we want to find is to find an alignment. So we want to try to match what they have in common. In this process of matching, the strings, what we do, we insert some spaces inside, so that the letters that look the same can be aligned in the same way. So what we do, we make an insertion here using this special character that is equivalent to do a deletion in the second string, and we end up with an alignment. Of course, this is not the only possible alignments. You can have many of them, and what you want to do is to find in somehow optimal alignment between those two strings. So we can try to assess the degree of correlation. Here correlation is not used in a mathematical sense, but relation, let's say, between those two strings. And you want to calculate a similarity or distance of those two strings, because you think that two strings that are very high similar are not far away, and two strings that show low similarity are far apart. So you want to understand how close are those two strings. So what you can use an editing distance. So we can compute a distance between those two strings, for example, using this kind of distance. And this is defined as the minimum number of operations, that is the insertion or the deletion or the substitution, to transform one string in another. For example, when we transform the first string in the second string, we need to insert one G here, and we need to replace a C with a T, and we need to erase one G. So the editing distance between those two strings is 3. The animation went the other way around. Okay, 1, 2, 3. So we can try to find a score of similarity. So we can try to assess the degree of similarity or distance between those two strings, assigning a score of alignment. And we can try to assign a different score for each of the possible situations that we have in the string. For example, we can give a score of +2 for each exact match, and a score of -1 for each mismatch or indel. So if there is a deletion or insertion, or there is a mismatch, we count for -1. If the two letters agree, then we count for 2. And we can sum then map, and we can get the score of this alignment. So in this specific case, we have 4 positions in which the letters agree, and we have other 4 positions in which there is either an insertion or a deletion and a mismatch. So the score for this alignment will be 4 times 2, that are the 4 equal, the 4 matches, 2 that is the score of each, plus 4 minus 1. So the score is 4. Of course, what happens is that you can assign different numbers, and of course, what you get is a different score, overall score. And what you want to do, you want to maximize this score. So you want to get something, you want to get an alignment, you want to find an alignment of those two strings in such a way that this score is maximum. So what you want to do is to try to maximize, this part, and to keep this part as little as possible. And this is what we are going to talk in the next part of the lectures. Thank you. So, as we said, now our interest is to try to understand how to find this scoring function. So we want to try to understand how we can measure the similarity of those strings that have been aligned. So this is we can define as the scoring function. So let's say that we have if X and Y are two single characters or spaces, we will denote with the sigma the score of alignment of X and Y. And this scoring function is sigma. And of course, we can build up different scoring functions. And during these lessons, we will see that somehow there are different ways to do this. And during the years, there was also an evolution in which those scoring functions were evaluated. We will not go in the detail on how the scoring functions were evaluated for the comparisons of amino acids, but we will see how we can use this scoring function in practice to find an alignment. So in case of amino acids, when we compare two amino acids, what we want to take into account is also the similarities from a chemical and physical point of view. So we will try to understand how we can take into account those similarities and those differences. So in case of pairwise alignment, we will call, let's say that S is a string, is a sequence and of symbols, and this is module of S is the length of S. And we will call SI the ith character in S. For example, if S is this string on a certain alphabet, then we have that the length of S is 6, and the third character in S is B. So if you have two sequences, an alignment A associates two sequences S prime and T prime to the two starting strings, adding some spaces, so adding some of those spaces in such a way that we end up with two strings that have the same length. So what we are doing in the end, we are comparing strings that are of the same length. And if we remove again the spaces, the special characters from S prime and T prime, we end up with ST again. Very simple. So we will say that the score of an alignment of a pair of sequences is given by the sum over the length of the sequence of those sigmas. So for every pair of character in S prime and T prime, we compute the score and then we sum up all of them. This is an L in this font. So the problem becomes the optimal alignment is the alignment that maximizes the similarity between the sequences or minimize their distance. So our objective will be to find this optimal alignment. And in the following, we will use the term score to indicate the degree of similarity between those sequences. Of course, the way in which we define sigma will make things different. So the way in which we count for the match and mismatch in insertion and deletion, of course, will make a difference in the optimal alignment. And the optimal alignment in this specific case has to be somehow checked by the biologist. So the biologist has to tell us if these alignments make sense from a biological point of view or it doesn't. So when we do the protein alignments, what we are doing, we are comparing strings, sequences of amino acids. As I said, those amino acids are 20 for a human being, and when we do the sequence alignment, we are also interested in understanding if there is some biological meaning in this alignment, or it's just a question. It's random in the sense that it's a question on how we choose the scoring function. And so we want to understand if this is biologically significant or not. As we have said, the scoring function associates a numerical value to each pair of characters. And so we will end up with a matrix in which we will have all the comparison between all the pairs of amino acids, and we will get a score from this. So this number will represent the degree of similarity of two different different amino acids. So there have been different proposals, and different matrices are used to compare amino acids. And the similarity of amino acids can be classified according to their physical and chemical properties. Somehow there are many different types of similarities that we can find. So we can cluster, let's say, we can cluster amino acid with respect to different properties, ending up in a number of classes that is different with respect to what we are taking into account. But still we need to take into account those properties. So now if we want to build up this scoring, matrices, then we have to build up those scoring matrices, we have to understand which are the amino acids that are similar and which are different. And to do this, we have to train somehow our algorithm on some databases. The first attempt to get these scoring matrices was done by Dioff in the 70s, and these are called PAM matrices. So what they were doing was they were actually looking at the mutations that were present in the families of protein sequences that were closer to each other, and they noticed that the replacement, so the substitutions that were among closely related sequences were not random. And so what they end up, they end up with the conclusion that the substitutions, there are some substitutions that are more easy than others, and this most probably means that these substitutions do not alter the protein structure and function. So this also means that homologous proteins do not necessarily need to have the same amino acids. So there might be different letters at the same position, but we end up with something from a molecule that is exactly the same from a functional point of view. So there are those PAM unit and matrices, and the PAM unit measures the distance between two amino acids sequences. So if we have two sequences S1 and S2, and two sequences are said to be at one PAM unit if S1 can be transformed in S2 with an average of one mutation every 100 amino acids. Of course, in the same position, what happens is that there can be several changes, and during these changes, sometimes the same letter can show up again. So we have the complete alphabet. These changes, if we fix the position, these letters varies, but it can come back. So this means that the difference between the two sequences that at one PAM unit of distance, it can be less than 1%. So this is identity. This is Italian word. So if a PAM unit is 0, then we have a 100% identity. So the two things are exactly the same. If we consider PAM 30, then we get a 75% identity. And so on. So the further we go, the less will be the overall identity. The fact that we can choose among different PAM matrices, and we will see that somehow they are all we can use different of those matrices. So each of these matrices is used to compare two sequences that are distant a certain number of PAM units from each other. So an entry ij of this PAM, for example, if we take PAM 120, each entry will contain a score that is assigned to the pair of amino acids AIAJ. And this score is proportional to the frequency with which we expect that IAJ replaces IE replaces IJ in two sequences that are distant 120 PAM units. And this is log normalized. So let's see which is the phase that those matrices have. So we will have, of course, they are symmetric, and somehow in every single cell we get a number that tells us which is this score when we substitute one amino acid with another. For example, if we have a 2 somewhere in this matrix, this corresponds to a value of .2 because all the values are multiplied by 10 to avoid the digital numbers. And the value .2 is the log of the relative expected value of the mutation. So the expected value will be 10 to the 0.2, that is 1.6. So we can recover somehow. We can use those numbers to compare those substitutions. More recently, let's say, there were also introduced some other matrices. These studies are like 40 years old. So somehow the quantity of data that were available was very limited, and so during the years there were more proposals for different matrices that might provide a score when we compare those pairs of amino acids. And the most recent are the Blosum that are somehow more used, are nowadays used in this string alignment algorithms. And what happens is that in the Blosum matrices we have blocks. The Blosum matrices are based on alignments of blocks of segments that are of closely related sequences. And in this work, it has been calculated the ratio between observed and expected amino acid on the basis of the observed sequences. So, so now we get somehow a different scoring matrices. And for example, if we consider Blosum 62, then since the sequences that we take into account are clustered, we are taking into account clusters that consist of sequences that have more than 62% of identity. So the higher is this number, the less divergent alignments we are looking for. So we are requiring to find alignments that have the letters more similar. Nowadays, Blosum is more used. So if you go, for example, to BLAST, that is one of these famous tools for sequence alignment, for searching for alignments in databases, then they use Blosum. There are some other tools that let you choose which one to use. With the nucleotides, the situation is much easier. So we do not say that a substitution is different. So if an A is substituted by C, G, T, then the score is always the same. So there is not this complexity. The complexity that we find in comparing amino acids is lost here. Although there are some functions in which the alignment score for an A and G is higher with respect to the other cases. So, but what we get is somehow a diagonal matrix. So we assign a score 1 if we match, and a score 0 if there is a mismatch. So now we need to understand how to treat the gaps. So we know how to treat the letters that we find in our sequences. Now we need to understand how to treat the gaps. So as we have seen, the two sequences, when we compare two sequences, when we find an alignment of two sequences, what can happen is that we have some bases, some letters that are exactly the same, and there are some other letters with a mismatch. And then we have also some insertion or deletion. So we need to add some spaces to one sequence or the other to make the alignment. So this, of course, we need to understand how to treat those gaps. And a sequence of continuous spaces is called actually gap. And somehow we need to find a scoring for this insertion. Of course, when you insert or you delete, equivalently, you delete in the other string a character, this needs to lower the score of the alignment. And we can do this. Actually, we need two scores. One regards the fact that you are actually inserting one gap. So the cost, you will have a cost that is related to the fact that you are putting an insertion. And another cost that you can take into account is the fact that you are adding some more gaps after you put a gap. This is due to the fact that when you start enlarging the space, so when you are putting gaps one after the other, what you would like to do is to try not to put too many gaps one after the other. So you do not want to produce you do not want to produce very large gaps. We are talking about global alignments. So we are talking about strings that are transformed in strings of the same length, and we want to align them globally. And actually, what happens is the GOP, so opening a penalty costs more than enlarging a penalty, but still enlarging a penalty has another cost. Inserting and deletion are so somehow when you insert the second character, this costs less than opening a gap. So let's make an example. So suppose that we have a certain sequence, and you want to align with another sequence. So when you open the gap, what you are doing, you are inserting this single space. And when you want to extend an existing gap, what you do, you enlarge this space, adding some more spaces to get the alignment. So when we do a brute force pairwise alignment. So the first idea that we can try to do is to try to do all the possible combination, all the possible alignments of those two strings when we are trying to compare. So we can, let's say that we have those two, that we want to align those two strings. We can represent this alignment as a single string. So what we do is we put when there are spaces, we put the letters, so we write AGA. Every time there is a match, we add the other letter from the other string that is matching or mismatching when there is a... So what happens? So now we end up with this is clear, right? So what we are doing, we are producing a single long string that is composed by the two strings. So now what we end up is with a string that has m plus n position if m and n is the length of the two strings. So what we are trying to do, every possible combination of this we can obtain actually if we it depends on the position that the letters will have in the string, given that they are always in the same order as they are in the original string. So the problem of aligning those two strings becomes select m position out of m plus n possible positions. And the complexity of this is of course very high. So even in the case of very small sequences, let's say you are comparing two sequences that have 300 positions, then you end up with a total number of possible combination that is very high. I didn't check this number. Maybe you would like to do. And this was published in Nature Methods a couple of years ago, this kind of thing. So, of course, this becomes, we cannot, of course, so even for small strings, even for small sequences, we need to find something else. And so the method that has been historically used for this kind of purposes is dynamic programming. This is a very funny word because somehow dynamic programming was invented by Bellman just to get money from the government. So it was looking for a fancy name for the technique that he had developed that could attract people from the American Congress. And so he decided that dynamic and programming were two words that everybody would understand and nobody would argue on. So, somehow there are, this dynamic programming is still present in some parts of softwares, even if we will see that somehow it's not very convenient for very large problems, but still it is still present in some of the softwares that we use today. So when we do an alignment by dynamic programming, we are trying to solve an optimization problem. So we want to solve an optimization problem, and we want a technique that somehow gives us all the possible, enumerates all the possible solutions, and it finds the best. So, in general, the objective, as we said, is to find an optimal alignment of S given some scoring function. So these algorithms are used to solve an optimization problem. In this case, we want to maximize the alignment score. So what happens is that this type of programming, of this type of algorithm, finds the best solution, breaking the original problem into sub-problems that are easier to solve. With respect to recursion, the difference is that we will store the partial results little by little in such a way that we can get the full picture at the end, and we do not need to recompute many times the same quantities. So, suppose that now we have two sequences, and suppose that we have that we want to align the first I character of P with the Q character, with the J character of Q. So what happens is that, suppose that we have already the alignment for the P that goes from 1 to I minus 1, and for Q that goes from I to J minus 1. So what happens is now that we have only 3 possible cases. So if we have already aligned the first part of, if we have already aligned the first part of the sequence, then what comes out is that we can try to match the I with J. Or another possibility is to insert, to align Q con J with a gap. Or the third possibility is that we can match the G with a gap. So if we have the solution for the initial problem, for the initial part, and we try to solve this new problem in which we have added another character, we can only end up in 3 possibilities. And we need to understand which is the best. So, okay, and so the first algorithm was introduced in the 1970s by Needleman and Wunsch, and And to compare the characters of the two strings, we use these scoring functions. So suppose that we are trying to compare the first character of S and T. What the possibilities are the three that we mentioned before. So either we just compare, we align the first character of S with T, or we insert a gap in the second, or we insert a gap in the first. So sorry, the other way around. Or we add a gap in the second or in the first, yes. And the action is associated with the score. So what we can do is we can use a matrices. This matrices somehow is a matrix in which in the first column there is the first string, the first sequence, and in the second there is the second sequence, and and we will fill this matrix line by line. The first thing that we need to do is to compute the first line and the first row and the first column. So since we are always comparing the character of S with a space, we will use the sigma score for this case. So in this particular example, we are using -1 for each of. We are using -1 when we compare a character with a gap. So -2, -3, -4, and so. And we fill up, and then we take every single and we try to assign the value of each cell based on this formula. So what we do, we take the maximum between the value that we have here. So if we arrive from, let's take this character, or any character inside the matrix. Maybe I have an animation on this. Yeah. Okay, so consider that we want to. So consider that we want to. We want to align that we arrived to a certain point and we want to compute the others. So at each step, what we want to do is to choose the maximum score to get aligning the character I with the character of J. So what happens is that we have three possibilities. So we can either go from here and we can try to match this character with this other, or we can match the I character of S with a gap, or we can match the TJ character with a gap, coming from either this direction or this direction. So if we say that we give a score of +2 if the two character matches, and we give a score of -1 in any other cases, then when we compute this V21, then we get -1 plus the 2, that is the fact that we are matching the 2. And or we have to find the maximum between those three quantities. So that are the three possible ways. So every time that we try to add a character to the previous problem, we have to understand if it is better to match the next character with a space or not. So in this case, the maximum that we get is 1, and it would be useful to record the fact that we obtained this one starting from this position and aligning the two characters that are equal. So when we do this process for every single cell in this matrix, we end up with all possible alignments of those two strings. So if we follow those paths inside this cell from the beginning to the end, we will end up with all the possible alignments. But the optimal alignments is obtained only if we look for a strategy, the one that enables us to get every time the best score. So what we are doing, somehow we have filled up the matrix and we need to come back again. As I said, we record the position from which we got the best alignment. It can be the case that the best score, it can be the case that the best score is obtained in two similar cases. So in two different cases can give the same number. So the maximum can be chosen in either way. So you have two possibilities that give rise to the same solution, as it is in this case. So these two could have been obtained either coming from this direction or coming from this direction. So what we need now is to trace back our matrix to get the optimal alignment. So, as we said, when we arrive at the end, we get the score of the alignment, and we need to do the alignment. What we do, we start from the end and we draw back to the beginning. So we will build up our alignment starting from the last letter and coming up on the strings. So, as it is, since the maximum here has been obtained in two possible ways, we can either choose to go up this way, or we can choose to go in this way. So once that we have aligned so we can go back either using the entry 6.4 or the entry 5.5. So I have an animation that shows this process. So let's suppose that we choose to go in this direction. So when we take this position, when we move on the left, what we are doing, we are aligning D with a gap. Then we do the alignment of B with B, and then we trace back the algorithm. So we align D by D. So moving along the diagonal makes we get the same. We align the letters. Either going in one direction or in the other, up or left, will introduce a gap either in one or in the other. Anytime we can choose. As we can see, we can have multiple possibilities, and in this case, we are choosing one of them. And finally, we get the alignment. So, if we were following the other way, we would end up with an alignment that has exactly the same score. But the alignment is somehow different. So now we need to decide which is the best alignment. So there are some techniques that enables us to understand which is the best. But somehow we have always to ask the biologist, the domain expert, which is the alignment that makes more sense on this. So, as we have seen, we need to build up this big matrix. So, the computational complexity of this algorithm, of course, will depend on the dimension of this matrix. So we can save some computations because we will use the same box, the same values over and over. For example, we will not need to recompute some of the values, and we can choose the strategy in which we fill up the matrix. In this case, I was showing I was going from top to down, from left to right. So the complexity this algorithm is quadratic in time. So if the two strings have length mn, then the complexity in time is of mn. But of course, if we can decrease this cost, provided that we lose some information. For example, if it is possible to implement the algorithm in linear space. This is described in a paper by Meyers, and somehow, if we want also the set of operations, if we want to store the operations that brought us to the alignment, then we need again quadratic space. In this paper by Ishberg, he showed how to produce an optimal conversion or alignment in linear space. Somehow, this paper was not taken into account by the papers that came after. So after that, this strategy was published, many people tried to decrease the space and the time complexity of those algorithms. And somehow there were a certain number of papers that were written. And but somehow they didn't look at this paper. In this here I put a link to some slides that graphically show how the algorithm works, and they might be interesting. There is also another paper that was published by Meyers that explains the idea that is beyond. Of course, you have to lose something. So if you do in audience space, you will get an optimal solution, one. When you do the dynamic programming, as we saw, there might be different optimal alignments, and you get them all. So you need to decide what you are interested in. You can also decide somehow, if you go, as we said, when you go along the diagonals of the matrices, when you go up backtrace along the diagonals, you get the alignments with the characters. So the alignment that you are looking for is an alignment that goes as much as possible along the diagonal. So you might decide only to look at a block matrix. So not to look at the complete matrix, but to decide to fix a certain K, that is the band of a diagonal matrix, and only look inside that band. So you only look at the solution that you can backtrace inside the band that is along the diagonal. This makes, of course, the computation faster, because you substitute one of the two and then with a K. So if K is small, of course, you can get you can get results much faster. Somehow here the problem is that if the two sequences are very similar, then you will find something in the between, no, in the center of the matrix. But if the sequences are very different, then you will find solutions that will be on the bands, on the outer parts of the matrix. So in the left down corner or in the right upper corner. But of course, somehow this might produce only the alignments that have no biological meaning. So if the two sequences have nothing in common, you get the alignment, you get the score, but this does not say anything. So reducing the complete matrix to a band, so computing all the steps of this algorithm on the band, on a central band, makes sense. It makes sense in this case. So, as we said, if we implement completely, so we have all the optimal alignments. But the number of those alignments can be very high. So we can have that in many possible ways we split. Now we have two possibilities that give us the same maximum score, and so we end up with a very large number of alignments that are optimal, These alignments might be insignificant for the biologist. And somehow what happens is also that a biologically meaningful arrangement may be a suboptimal alignment. So what the biologist might ask is not a perfect, an optimal solution, but a suboptimal solution, because this, as we said, depends on the quantities that we decide to use in the scoring functions. Okay. So what we have been doing till now, we have been trying to take two strings and align them on the wool length. What we are now trying to do, what we would like to do, is to see if some substrings, some parts of those two strings really match. So what we might be interested in is to see if there are some local places in which the two strings match, because this might provide some information, again, useful from a biological point of view. So if you have some places that are very, very similar, this might provide some hints. For example, we could find we could find a new gene, or we could find a specific part of the genome that have a biological meaning. So our interest now switches from the search of global alignments to the recognition of local regions with high similarities. And for example, we can use a short sequence with respect to a very long one, trying to match a particular specific part of this long string, of this long sequence. Of course, if you look at the global alignments, what happens is that you will find the small one split around. Now what you want to do is to try to keep the small sequence together, and to try to find if there are regions in the long strings that are very much similar. Somehow we can still use the previous algorithm, but we need to do some modification, and those modifications were introduced by Sweet and Waterman. So the modifications are somehow very, very simple. The first thing to do is you need to change the initialization of the array. As we saw, the first row and the first columns of the complete matrix had, if we score -1. If we start from -1, then we have -1, -2, -3, and so on. So what happens is that going in one direction, so adding spaces in one of the sequence or in the other, becomes very, very costly. So if we put 0 instead of these numbers, then moving around moving the short sequence around the long sequence becomes much easier. It becomes less expensive, right? So the first thing that we need to do is to set the first row and the first column are set to 0. And another modification regards the filling of the matrix. As we have seen, when we build up the matrix, in any case we need to choose the correspondence, the insertion, or the deletion. And even if the overall, even if any of these choices will decrease the overall score along the path. With the Smith-Waterman method, we can stop in any place we like. So the alignments, when we backtrace the alignments will not go from one end to the other end of the matrix, but it can be just a small piece of the backtrace inside the matrix. So what we are doing, we are just taking a small pieces and we are trying to locally align with another piece. So now the last problem, the last thing that we want to address is in 5 minutes is how we can decide if in the matching there is a real relation, and we can do this from a statistical point of view. So what we can do, we can try to compute a number that tells us how good is this alignment with how good is this alignment with respect to some random alignment, or we can try to see in a control population what happens we can see in the control population what should be similar. And we will only cover three very simple statistics. And we will use very and we will use, for example, a zeta score. So let's say that we have multiple samples or tests, and we can compute average, and we can use average and standard and mean, and we can normalize, and standard deviation, I'm sorry, and we can compute how far is this score that we got from the average score in the unit of standard deviation. So we can decide using Chebyshev theorem that if this score is 3 units away from the average score that we get, then this score is useless. This alignment is useless. Or we can try to use a p-value. So a p-value is the probability that this alignment. Okay, I finish this slide and we go. So we can use p-value. So we do an hypothesis test, and we try to understand which is the significance of the test. So what you are seeing, what you are trying to see is that the alignment that you found is not trivial, is not obtained by chance, which is the probability that the alignment that you got is not taken by chance. So we will end up this block here. And till now we have seen these two algorithms that are used, these dynamic programming algorithms that have been used over the years to align, to do the comparison of two sequences. In the next block we will see the multiples alignments. So we will see how to compare multiple strings all together in one shot, let's say, a certain number of heuristics that are used to this purpose. And then we will see those new, let's say, these algorithms that have been produced in 2009 and are still used to align sequences in the case of next generation sequencing technologies. Thank you.